We're really happy that, again, that Jonathan stepped up here. He's an integrated ecosystem assessment research assistant in the Ecosystem Sciences Division in the NOAA Pacific Islands Fisheries Science Center. He has a PhD from right here at UH and a BA in zoology from Prescott College in Arizona. He's a project lead for the West Hawaii Ichthyoplankton Project, the SLICS project, and the West Hawaii Integrated Ecosystem Assessment Project with JIMAR in the Science Center. His talk is entitled Ecological Interactions in Larval Fish Nurseries from Microscopic to Ecosystem Scales. And I'll ask the speakers, if you would, please be sure when people ask questions that you reiterate the question, because there are some people that are viewing online and they said they can't hear what some of the audience is asking. Thanks.
Yeah, don't wander away from the microphone.
Fair enough. I'll stay here.
And Doug's keeping the time. He's going to be really strict.
Yeah.
Got it. Check. Rolling. Thanks for making it back from lunch, everybody. Yeah, and if you're just walking in, I'm not Greg Asner, but we do have remote sensing generated from him, and coral reefs are relevant, so it's pretty much the same thing, right?
Without all the awards.
Yeah, without, whatever. Who needs all that stuff? No TED Talk yet, but. So this is a very collaborative project, and just want to acknowledge all the people that have worked on this. It's been growing and still ever growing, and includes a lot of JIMAR support, and of course was funded by the JIMAR Postdoctoral Fellowship. So it's really, project may not have happened without that support. So I want to thank you guys up front. And jump right in. So life for most marine fishes begins at the surface. So buoyant eggs are spawned, they float. Within about a day or two, they hatch into little larvae, and then for weeks to months, they're developing and dispersing out into the open ocean before they transition into juveniles and then settle back to the reef or wherever adult habitats they come from. And larval fish are foundational for ecosystem structure. They're the next cohort of fish that we're managing and eating, and they're trophic links for tons of things in the pelagic as well as coral reef and mesopelagic ecosystems. So they're really important, but we know very little about what happens in this stage. Essentially the ecology of larvae, where they go, what they eat, and how they're impacted by human-induced stressors during this really vulnerable life stage. And so really a big goal of this project was to try to identify essential early fish habitat, which is also a big goal of ecosystems-based fisheries management. And so really we started out was where to find all the baby fish, which brings us to slicks. So slicks are these ocean surface feature. They're ubiquitous in coastal areas around the world, and in some places, they've been mostly studied in temperate, have been shown to accumulate a lot of fish and plankton and debris. And here in Hawaii, and particularly in West Hawaii, these are a dominant surface feature. So you can look out on the water surface pretty much any day, you can see these slicks. If you've flown over, actually flying, leaving any of the airports in Hawaii, you've probably seen these in most coastal locations. And satellite imagery shows how ubiquitous these are, and I'll get into this a little bit later. So these are a dominant surface feature known to accumulate plankton in other places, but they hadn't really been studied here. So this was a key place to look. And then this time lapse shows how dynamic these features are. This is a whole day in 10 seconds, but showing how slicks are coming and going. They're propagating towards shore. They're moving along shore. So they're really dynamic habitats, not static at all. So we did 3 research expeditions to try to find out what's in these things and to really characterize the community and their impacts on this ecosystem. So we did a lot of new spawn tows, both in surface slicks and then in ambient water, essentially right outside of slicks as a baseline. We did most of it from small boats, but also sampled off of the SETI and surveyed most of the West Hawaii coastline. And then the very non-tech savvy part of it was all the laborious manual sortings. This is pretty old school. We wanted there to be those advanced new tech approaches to this, but it's still not there yet with the automation and the AI and image analysis and all that. We'll get there soon, hopefully, but this was all done with humans under the microscope, thousands of pieces sorting. Emily Contreras, who did a lot of it, is here today. And so really a lot of work goes into, and a small army of people goes into processing all these. So what we found is that slicks accumulate all kinds of plankton and debris. So this is a plot we'll see a lot, so I'll explain it here. So this is the ratio of mean densities in slicks to that in ambient water, and this is the mean with the 95% confidence interval. So this shows that we have, on average, 2 times as much phytoplankton, 4 times as much zooplankton, 8 times as many larval fish in slicks as in outside ambient water. And we also quantified the amount of debris. And this is using dry weight. Organic debris is all the leaves, twigs, arthropod molts. Essentially, when you're small, any little bit of habitat is potentially structure and shelter. So we wanted to quantify that. But unfortunately, also a lot of plastic debris too. So about 30 times the amount of organic debris and more than 250 times the amount of plastic debris if you use weight. If you use density, it's about 125 times as much. So either way you look at it, a lot of plastic in these habitats, which we'll get to. So really what this looks like is that slicks concentrate all of these buoyant and upward swimming animals at the surface, and the convergent flow that drives what makes slicks, which are driven by mostly internal waves, but also other fronts, tidal mixing fronts and headland fronts. Essentially the same kind of convergent pattern on the surface that brings all of these organisms and debris together. And so all their ecology is really densely concentrated into these habitats. So let's take a look at the taxa that we find in them. So we find really dense and diverse zooplankton. So these are all the holoplankton or the non-larval invertebrates. And 9 out of 10 groups are at least 2 times as abundant in slicks as outside. And some of them are more than an order of magnitude more abundant. So really most of the food source for larval fish are concentrated into these habitats. And also similar with larval invertebrates, nudibranchs, shrimp larvae, stomatopods, crab larvae, all highly concentrated, at least 5 times more abundant in these habitats. So really across the board, these features are affecting essentially the entire Neustonic system. So next, the fish diversity. So we identified more than 13,000 individual fish and found over in slicks more than 118 species from 55 families. So a huge diversity representing roughly 10% of all fishes found in Hawaii are being hosted by these habitats. And a huge mix of things, everything from flying fish, mahi, spearfish, tons of different jacks, reef fish, damsels, chubs, other jacks, triggerfish, filefish, and even really deep things like anglerfish, things from the mesopelagic, that during these first weeks to months of their lives are all converging and living in the same habitat at the surface, which is the only time in their lives where they'll share that habitat. So not just more presence and diversity, but also more abundance. So these are the 33 families that were relatively common, present in at least 4 toes. And 90% of the families that we find in this community are at least twice as abundant in slicks than not. And these guys over here that are more than 25 to 100 times more abundant are essentially only found in slicks. It's their critical early life history habitat. Things like needlefish, every species of locally occurring flying fish, this is pretty much the only place that they're found. And this is a mix of reef-associated pelagic and mesopelagic taxa. At least 50 species of reef fishes, 30 species of epipelagics, and about 20 species of mesopelagics. So really an incredible assemblage of biodiversity that I can't really find another example where animals from such disparate habitats are sharing the same nursery habitat. It's really quite unique. And not just more fishes, but also bigger fishes. So this is the size distribution of larvae in slicks in gray and outside in blue. And we find more and also larger size classes inside, which we are taking as evidence of potential habitat selection. So when we map or model the probability of occurrence in slicks goes up as fish get older. So as they get older, they become more developed, better swimmers, and we kind of assume are capable of choosing where they want to be. Late larval and juvenile stages are pretty nectonic by that point. So we think this is good evidence that the larger ones are at least actively seeking out these habitats. So what does this look like from an ecosystem perspective? This is a graphic that we're trying to kind of represent and working on. So if you have any suggestions, I'm open to them. We haven't submitted this yet. But all kinds of things that we just talked about are accumulating inside of slicks, illustrated here. And then those larvae are then propagating and radiating out and replenishing adult populations in coral reef, shallow coral reef, open ocean, and even mesopelagic, and even down into deep sea and the benthic. We get larval tripod fish that adults are at 3,000 meters plus, larvae living up at the surface, bringing energy down with them. So these fishes are playing important roles on coral reefs. We have several species of jacks, large caranx, predators that, of course, control a lot of the reef structure. We have lots of blennies and gobies that are providing a major food source for a lot of reef organisms, as well as the benthic macrocrustaceans, shrimps, stomatopods, and crabs that are also a huge food source for a bunch of coral reef animals. And then also out in the pelagic realm, flying fish, for example, are huge prey items for most pelagic predators. So billfish, mahi, sharks, as well as most of Hawaiian seabirds are relying on flying fish as a dominant part of their prey. So where these larvae then go, develop into adults, are potentially having profound impacts on food webs in multiple different ecosystems. But as we talked about earlier, it's not all good. These habitats also accumulate hazardous amounts of potentially toxin-ridden plastics. This is from one 7-minute tow in Kiholo Bay, a couple hundred meters from shore, and there's 37,000 pieces of plastic in this image that was from one tow. Most of them are the size of tiny sand grains. So we find that these features really dramatically increase the amount of overlap between fish and plastics to a really alarming level. So we find in ambient water, fish outnumber plastics, but inside slicks, which is where most of them are, they're outnumbered by plastics by about 7-1. So really, they're highly correlated. Where we find more plastics, we find more fish. And so the potential interactions with these pollutants are becoming exasperated. So this led us to worry if they were consuming plastics, which unfortunately we found that they were. This was the 6th fish that we cut into, had this tiny piece of what looks like polystyrene that was virtually invisible to naked eye without looking at it under the scope. And we dissected over 600 little larvae. The smallest we could get was about 1 centimeter, and we found them in every size class that we looked at. Most of them were these microfibers, really tiny microscopic microfibers. And there was a high prevalence of blue. There's a lot of blue pieces that we're finding, which is an interesting pattern. Not all taxa respond the same. We looked in 9 families and found plastic congestion in larvae in 8 of them. Goatfish were the only one holding it down, not eating plastic. So whatever they're doing it right. Mahi were also really good. We only had one individual that got, or 1% that got those, so they're also better. But fish that we eat, swordfish, spearfish and marlin, about 10%. Flying fish is the highest one that we found, which is potentially really alarming because of what we just talked about. They're an integral part of this pelagic ecosystem. So we don't yet know what the impacts are. Still very early in the science. It's probably not good. They could die from eating a piece. They could just poop it out. But the possibility of bioaccumulation is definitely real when they're eating plastics when they're less than a week old. So the next steps to this were really to take these in situ observations and try to scale them up to the ecosystem level so we can really measure what the impact are on the whole ecosystem. So this is what led us down a long road of synthetic aperture radar images, drones, and remote sensing to try to figure out how to map these features. And so for the first time, we've been able to map slicks from space using the satellite images from the Dove constellation, which is the same as the one used for the Allen Coral Atlas. So these little nanosatellites up in orbit. And Greg's group, specifically G-Way, figured out how to use AI to recognize using glint and the same features that we use to see them on the surface, to pull them out and then essentially create maps of those. So this was a huge advancement that we could then quantify the amount of habitat at an ecosystem level. So bringing that down to just one day as an example, we found that on average, slicks cover about 8% of the surface area in our 1,000 kilometer study area, which is that bound there. But they contain 43% of all the larval fish, and this percentage goes up the older they get. For pelagic juveniles, it's about 90%, and for some taxa, it's all of them. They're only found there. And about a quarter of all the zooplankton prey at the surface is concentrated here. So dense food source, great place to be. Also good for debris because you have some habitat structure. They're basically little fads, but obviously that's a double-edged sword with plastic debris because they're being not just surrounded by plastics that they could eat, but also surrounded by potentially toxin plastics that they may be absorbing toxins from just being around them. So the impacts are still really unknown, but a lot of good and some bad. And then some of what we're working with now is to try to, because satellite images are still just snapshots, we want to try to scale up and look at how these mechanisms are being driven, what are their underlying physics, and how that changes through space and time. So we're starting experimenting now with coupling these time-lapse cameras with in situ instruments. They're looking at acoustics, both current profiling and backscatter. This is backscatter here, so essentially like an upward-looking fish finder. The mooring is this little purple dot here, which is in 600 feet of water, by the way, in middle of the E. That's right next to shore. And we can see the slicks forming there. And probably they're driven by a variety of mechanisms all within the same day. So these kind of look more like tidal mixing fronts, accumulating and aggregating, all kinds of things, including debris and plankton at the surface. And then a set of internal waves coming and structuring that, and then bringing everything that's accumulating slightly offshore. And now, as we're going to see now, is actually delivering that to reefs, bringing pelagic subsidies of food and zooplankton, which we've seen in the field, and also potentially delivering larvae back to the coral reefs. And so this shows huge promise for giving us a more synoptic view and a quantifiable view that we can incorporate into fisheries and ecosystem models. And this camera system is taking an image every day for a year, and it's out continuously now. So we're going to learn a lot in the next year. And then the last way that we're trying to expand the scope of what we're able to look at is using samples that were archived at the Science Center. These were Newston toes that Bob Humphreys did for the Life History program, biosampling looking for commercial fishes, which they took out and kept the rest and preserved it, luckily. So we had more than 600 toes going back to the late 90s that we've delved into. We've already processed 3/4 of it and have really now an amazing data set that we can look at not just our samples, which are the blue ones that we did recently, but a whole slew, and really characterize these, not just these habitats, but also the entire ichthyoplankton community and how it impacts ecosystems. And so that's what we're working on now. And I just want to end there by saying, first, thanks to Mark Merrifield, which I hope is watching from home, and Doug Luther for funding the fellowship and supporting this project. Without it, it would not have happened, particularly the JIMAR Council, also Jeff Drazen and other people at SOAS that have been very supportive, as well as tons of people who have helped through various aspects. So thank you. Questions? Jeff?
Been able to analyze the beam flow of the slicks, and is there generally a shoreward progression or generally an offshore progression?
Great question. So have we been able to measure essentially the velocity and shoreward progression of slicks? And yes, for the ones that we've measured, there's on average a shoreward progression, but some of them moved offshore, which was interesting. We had to double check those measurements, make sure we got it right. But yeah, most of them, particularly the ones driven by internal waves, are propagating towards shore, but about 10% of them are kind of sloughing back. Joe.
So you said lots to eat, great place to be. So why don't we see an accumulation of the things that are eating the things in the slicks, in and around slicks?
Yeah, we do.
Repeat the question.
Yeah, sorry. Yeah, essentially, what else is accumulating in slicks, particularly the predators? And yeah, so that's one aspect that we haven't been able to measure because we looked at all the small stuff. But fish, large schooling pelagics are definitely there. We see schools of aku that are popping in and out, even dolphins that are popping in and out of slicks. And then the fishermen troll in them all the time. So they know that they're going to get more likely hits there. So that's, yeah, one aspect that we want to try to quantify is what the predation rates are. But these things are definitely attracting more predators for sure.
Okay, thanks again, John.